excited to introduce our second speaker and our second last speaker of today, Dr. Dylan Cooke, who is here with us from also from SFU, and is really interested, the director of the Sensorimotor Neuroplasticity Lab, and is really interested in the intersection of brain organization, the environment, and behavior. And we're going to hear about that today. I did a bit of internet sleuthing last night and discovered that Dr. Cooke, who is clearly a brilliant scientist today, has been experimenting for decades and was experimenting in his backyard as a young child. So with that, I'm going to turn it over to you, and I'm really excited about your talk.
Thank you. I wonder what website that was on. Okay, so I'm going to define brain dynamics very broadly. And today I'm going to be talking about brain dynamics across timescales, from brain evolution to developmental plasticity, adult plasticity to patterns of neural activity. And these are arranged in terms from long to shorter timescales. And in this framework I'm introducing, each one is constraining the one below it. And then also, in the context of this framework, I'm going to be talking about brain maps and individual variation. And I hope a lot of this is going to be familiar to you, but my point is the framework and how things are related. So let's start with brain evolution. For some perspective, this is a tree of life showing that most of life gets along just fine without a brain, and that we animals are the exception. We do have brains, all these others do not. So zooming in on animals, most animals do have brains. There are some exceptions. Animal brains have been evolving for about 500 million years. There's a tremendous variability in these brains. Many of us here are focused on vertebrates. My research has been specifically on mammals. And so zooming in more on mammals, all mammalian brains share a basic architecture, but more than 200 million years of mammalian brain evolution has produced a lot of a range of properties, brain sizes ranging by 5 orders of magnitude, very different shapes. Some brains have cells gyri, some do not. The number and density of neurons varies. The number of cortical fields varies. And so one example factor that is largely determined by evolutionary genetics is the relative sizes of sensory domains in the cortex. So each of these is one hemisphere of animal, and it's just schematically showing the relative proportion of the cortex that is devoted to processing different sensory domains across 3 species. And so the squirrel's brain is adapted to visually guided behavior, and the duck-billed platypus is very much emphasizes somatosensory processing, and the ghost bat, which is echolocating, is very much auditory. So this is going to partly constrain the way each animal develops, learns, and behaves. So a homunculus is an example of a brain map. And in a brain map, related information is processed together. So in this case, it's adjacent places on the skin tend to be processed near each other. And so you get somatotopy, or in the visual system, you get retinotopy. The other thing to point out here is cortical magnification within the maps. So in humans, the lips and hand are overrepresented relative to other body parts. And then, of course, across different animals with different important effectors, different ways that they interact with the environment, their brains emphasize different body parts within the somatosensory. So the duck-billed platypus, the bill is very much over enlarged, and the star-nosed mole, and the hand of the raccoon, and so on. And the raccoon hand is so specialized that finger on the raccoon's hand gets its own gyrus on the brain. So, these animals' brains have, of course, specialized for the environment that they developed in, and evolution has selected for that. So, brain evolution constrains how sensory input, behavior shape the development of the brain. But the input that animals get during critical periods also can have a huge effect on the overall organization of the brain. So normal development requires normal sensory input. And an example is that if you mess with this, if you change this, for example, if you remove the eyes of a possum 4 days after birth, then the visual domain of the opossum is entirely taken over by and of course, famous sort of Hubel and Weasel work. If you manipulate the characteristics of visual inputs, for example, you get permanent functional changes, even including blindness in some cases. But the key thing is that this has to happen within a critical period. And if the hello, hello. Should I stay near the lectern? Yeah, okay. In this example, a monkey was trained for thousands of trials to detect a small change in vibration applied to one finger. This changed the cortical map, but most of the change was within the representation of the finger that was trained. It wasn't across the other fingers. It wasn't across the rest of the body representation. It wasn't across different sensory modalities. It was very restricted. This particular image shows the changes in the receptive fields in the hand, but there's a similar kind of restriction that happens in the changes in the brain. So adult experience can change maps, but it's mostly within subregions of the maps that are originally defined during the larger scale evolutionary and developmental changes that are processes that happen. So in other words, developmental plasticity is constraining the range of what adult plasticity can do. And so this is an example of sensory plasticity. I want to make a different point with this example of motor plasticity. So this is a classic study by Randy Nudo and Mike Merznick, in which they're training monkeys to pick up a small food pellet in increasingly small food wells. So it gets more and more difficult. And before and after training, they are creating motor maps, moving electrodes around the brain, around the motor cortex, these animals, and electrically stimulating and evoking movements. And they are focusing on part of the map that's evoking the movements that are used in this trained task. And a lot of these relevant movements, the representations in the brain expanded after training. They found that this particular combination of finger flexion and wrist extension, which the monkey does just at this crucial moment when it's picking up the pellet, that this was the one of the largest changes that they saw. And so that's schematized in this Venn diagram right here. That expanded the most. They also tracked over cycles of learning and then extinction and learning and extinction, how the animal's performance changed. And then they also tracked how this particular representation of this crucial risk plus digit representation changed in size in the brain, and they tracked each other quite nicely. So so this and other studies suggest that adult plasticity that reorganizes this map, it is constraining the neural activity that's driving this optimal behavior so that it can accomplish this difficult task. So I'm going to switch gears now a little bit. A number of the talks, including Dan's talk, which is a nice segue, have touched on individual variation between humans, within the human species, across individuals. There's clearly, we are individuals, our brains are different. Lab animals also are individuals when it comes to their brain organization. And this seems to be particularly the case in motor cortex. So it may be partly the way that motor cortex develops and the sort of competing factors that shape its organization. But there's a tremendous amount of variation in motor cortex. So this is an early paper showing the motor map of a rat. And now, I was going to say no one highlights variation in data, a laudable exception. But generally, I think the thought is like, if there's variation in your data, then you're sloppy. And indeed, this 1986 paper, the authors, there's one sentence on page 18. Oh, by the way, these maps are reviewed. But you have to take these maps from different parts of the paper and put them next to each other and color code them to actually see how crazily different they are. This is the representation across 3 animals, 1, 2, 3, of just the forelimb motor representation. And the colors are all of these different component forelimb movements. Change this. Let's see. Let's see that on there. Oh, I've got to do this. This is fun. Okay, I can point like this, but not on here. Yes. Oh man. Okay. Exactly. So the colors are all these different movements of the forelimb. And it's just amazing how different these are. These are all animals raised in little shoebox cages in the same lab, and their forelimb representation is so different. So what's going on here? So normal development may produce a wide range of motor organizations. As I said, this may be just because of all the competing factors that shape the topography of motor cortex. And then adult plasticity is going to produce smaller changes, but because of this starting large variation from developmental plasticity, the result is this wide range and variation of organization of the motor cortex. The variation could also be genetic as well, especially if you're not talking about inbred mice. And so, there could also be variation just based on the genetics. But the question is, how might this map variability affect neural activity and behavior? So this is a question that my lab is beginning to approach. But before I get to that, I'm just going to back up a little bit and talk about some previous work I did in which I showed that if you electrically stimulate the brains of monkeys with certain parameters, you can evoke a range of movements that look a lot like natural behaviors. So you can evoke defensive movements, and you can evoke climbing and leaping postures, and manipulation, and feeding, hand-to-mouth movements like this. And a lot of other labs have shown this in other species. One of the important things that we showed was that if you took the domain in motor cortex, where when you stimulated, you evoke defensive movements, flinch movements. If you then infused GABAergic drugs in there, you could turn up the defensive response to a real stimulus, an air puff. You turn it up and down selectively, but not affect other behaviors that use the same muscles. So it seems like this spot in cortex actually has a role in controlling a natural sensory motor response. Now, one of the key aspects of these complex movements is that it's all about coordinating muscles, not just, for example, within the forelimb, but across the body. You know, these flinches are sometimes like, you know, a movement of the arm out and the head turning and the face flinching like this. So it's not surprising that there's somewhere in the brain that does something so important. But the nice thing about it being in motor cortex is that it's relatively accessible to sort of study at a very granular level. And so that's one of the things that we want to do, a sort of elemental level. So the questions that we're wanting to ask are about the cortical control of muscle coordination, and then using that as a model for individual variation in brain organization. This is our basic preparation. I'm not going to go into the technical details. The basic idea is that we are moving an electrode around in the brain to one spot after another and electrically stimulating to evoke movements while we are recording up to 16 muscles in the brain. We're also analyzing the EMG that we get in real time so that we can control when we stimulate and avoid times when there are spontaneous muscle activity that would contaminate our stimulation evoked muscle activity. And we create two kinds of maps. One of them I call a kinematic map. Essentially, this is us observing the movements that happen during stimulation and videotaping them. And the striped colors indicate the combinations of body parts that that move when we stimulate a range of different sites. And I'll note also that we get these maps across motor cortex and somatosensory cortex and anteparietal cortex. We also create these EMG maps that are, and that's what's on the right here. These are heat maps of muscle excitability at each of the sites that we stimulate. So each of these little maps is the heat map for a single muscle. And so we can look at individual muscles, and we can also look at combinations, any combination of muscles. So this is the representation of the coordination of these two muscles here, and these two muscles here, or three muscles here. And in addition to this, we also, at each site, we have, of course, the EMG data, the temporal data that we've recorded, which sometimes has sequential information. Will sometimes evoke movements like running or digging movements that have more temporal structure. So it's quite a rich data set compared to just the kinematic map, which is just about body parts. So our goal is to describe the full variability across individuals. And then we want to see if this correlates with behavior or learning or rehabilitation. And we can ask, does this individual variation, does it constrain behavior or learning? Can we predict behavior or learning? That's the direction that we're heading with this. We have, this is data from last year. We only had 3 muscles. Now we're getting 16 muscles routinely. And so our students this week are analyzing that data. And I'm hoping that I'll have a lot more to say about that soon. But anyway, thank you for your attention. And I look forward to questions.
Thank you, Dylan, for such an interesting talk. We're going to open up the floor for some questions. Does anybody, let me just check the Zoom because I see there's actually a couple questions there. Yes. Okay. Yes, we solved that problem. What are your questions, everybody? Open up the floor. We are. You're still on Zoom. We're just not sharing the slides.
We can just move that over.
Here we go. Yes. Okay. And I'm going to move out of the way because we're sharing this guy.
Ready? Sure.
Wondering if you have any ideas of how you're able to are they fairly stable across days, or are they vary?
Yes, yeah, they're pretty stable across days. There are definitely small changes that happen. There have been experiments where whether we're recording sensory responses or doing electrical stimulation, that you will see some small changes across time, but they're pretty stable.
And if there's any kind of learning,
I should say that's assuming you're not doing some learning paradigm.
I'm trying to think in the context of things like, for example, TMS, where you're trying to do focal stimulation. I know, TMS, some of the initial things for calibration to get a motor response, TMS, and assume that stimulation would be the same for other parts of the brain. But if there's variations in terms of representations of an area, whether that would have implications for how you would then target stimulation across days, for example. I mean, there might be slight changes in representations.
Yeah, I mean, I would think that the scale of the changes across time, barring some sort of major learning paradigm, would be relatively small compared to the scale of EMS stimulation.
Do you have any thoughts on that? Or just, since you're one of the TMS people in the audience.
I would agree with the response. We tend to think about TMS as focal, and while it's true that it's compared to other neurostimulation techniques, it's more focal. It's still a relatively large area compared to those targets that you're aiming at.
What's your resolution?
Quarter of a dollar coin.
Okay. Yeah.
In brain size, that's a lot of neurons.
Even in a human.
True in a damaged brain. In fact, it's not stroke. So patients who don't have an MEP, and then it will emerge.
Oh, yeah.
With interventions and with just time.
I mean, I've seen just an anecdote relevant to that. Years ago, we were trying to do a lesion study in monkeys. They were lesioning with your private cortex. And you do the lesion, and then it's a behavioral study. And so you have to wait a couple of days and be off its meds so that it's willing to actually do the behavior. And then we got one interesting day of data where the results were different on this behavioral task, and then it was completely back wrong. Very frustrating considering how much work had gone into training that animal into a lesion. But I take your point.
But if it's a human stroke patient, that's fantastic.
Right. Yeah, it's good news for the monkey and the human.
Yeah.
Bad news for the experimentalists.
And then just on the other side, I mean, you started off with a very good high with evolutionary changes, organization of the brain, the existence of the brain per se, etc. And also you started tying it into plasticity. And I wonder if across these different species or different preparations, if there's also variations in the degree of plasticity in these systems as well.
I think I don't know, and my guess is that that's kind of a hard question to answer, just because often when you're doing plasticity, you're talking about training an animal, and it takes a long time to figure out how to train an animal of a particular species to see what sort of, I mean, even to learn how to train an individual, especially when you're talking about something like a monkey, you have to figure out what drives them. Are they food motivated? Are they thirst motivated? Are they motivated by social interactions?
Yeah.
And so that I would guess that a bigger factor is going to be their sort of trainability, and that's going to maybe mask the
Yeah, just
fundamental differences in plasticity.
I mean, you can think about that in the context of also the pressures, as well as whether you're in a volatile environment or not, which with plasticity would be helpful. And this is a supposition, I have no idea whether that might, versus an environment where it's fairly stable, plasticity would actually not be a good thing. I mean, there's examples that I forgot what species it is that eats its own brain after it finds a rock to sit on. Yeah, I'm good. I'm happy to eat now. Eats his brain. It's
Great.
kind of what happens when you get tenure.
Looking forward to it. So, even though animals have very different sort of selection pressures in terms of their behavior, I think a lot of this plasticity is sort of baked into the developmental plasticity that happens. Right? That there's this like competition for real estate in the brain that is very intense during development, so that if you suddenly have one eye that is closed during a critical period, then other senses or inputs take over that. That's still going on later on. I mean, when you have a major disruption as an adult, you can still get smaller but relatively big changes. And I think that's probably because that's the way that evolution has decided to build brains. I can almost call it like an epiphenomenon of the way rather than something that is, it definitely can be adaptive for learning, but I think it may have been the brains that speculation.
Yeah.
Lena.
I have a quick question about these mention of the spontaneous activations in the cortex. So you try to not stimulate during to avoid noise, but I'm wondering if you or anyone else has tried to think of these as potential phenomena of interest, especially it relates to noise in the brain is a beneficial component. And to go beyond that, I'm wondering if anyone's looked at them, whether there's any signature, whether they in some way they mirror the the activations that occur during stimulations, and whether there's any individual differences.
So a little more background on that. So the spontaneous movements that we see are in the context of the fact these are anesthetized animals, but they're lightly anesthetized with ketamine xylazine. If you use other kinds of anesthesia, you don't get spontaneous movements, but you also can't evoke movements. So there's a reason that this is one of the anesthetic preps that allow you to do stimulation of movements. There's other kinds of anesthesia like isoflurane gas that is great for recording from single neurons. And it's nice because on the animal is, of course, is not moving around at all. It doesn't do anything. That's not true. It does less to neural responses, but it just wipes out stimulation movements. Right? So then the question is, is this spontaneous activity during anesthesia a meaningful signal? I guess I'm mostly just frustrated by it, although we use it kind of as a proxy to know is the animal at the appropriate anesthetic level. So even with ketamine xylazine, if the animal is too deep, then you're going to get less or no stimulation of evoked movements. So is it relevant? It's definitely related. If you stimulate and evoke a particular movement, what we've seen is we will see sometimes spontaneous movement continue in a similar part of the body. Not like a seizure, but just if there's more baseline spontaneous activity happening there.
I mean, I ask this question because there's tons of research looking at hippocampal stimulation, but also recordings of these waves that get spread out of the hippocampus into the cortex, and they instantiate some kind of states that have been linked to mental replay or sometimes replay of future things that might be explored. And interestingly, they have been noted to occur under states that are usually of inactivity or restfulness, which may be somewhat towards along the continuum towards something. It's not an anesthesia, but it's a restful state. So it just occurs to me that what you're seeing, it just might be that what you see in these spontaneous activations might be some kind of a replay or pre-play or some sort of a simulation that the motor cortex does by virtue of, I don't know what other regions of the brain might trigger it to do it, but it might be a part of the learning and the plasticity. So that's why I'm asking about it.
Yeah, could. I think you would, I think it would be more relevant if the animal wasn't anesthetized. And you can do preps where you're stimulating awake animals. The challenge then is that they're sometimes responding. They are behaving. We're often happy by feeding them food, and so they're thinking about reaching out and trying to get raises the monkey. But I think you could look at spontaneous movements that were not those are clearly behaviorally directed. One step that would be more relevant to regular brain function than in an anesthetized brain. My speculation.
Thank you for a great talk, Dylan. I was interested in that slide where you have the tree of life for brains, and then you noted the variability in different properties of the brain. So for example, it was smoothness versus convolution or size. So for those properties, is it known whether there's selection, whether the variation is driven by selection or simply genetic drift? So I imagine it might not be the case that all properties or all of those properties are driven by selection, but maybe some are. And also, does your experimental paradigms or do your experimental paradigms provide insight on that?
So to the first question, brains are so expensive metabolically, my guess is that it's mostly driven by selection. And so there's a huge literature on this, and just taking the example of magnification, brains that are flat versus have cells and gyri. One of the things that gives you is just more cortex. And so in certain lineages of animals, there's a trend toward more encephalization, more growth of brain relative to body size. And often in those cases, you will see gyrification, just so you can fit more cortex into a brain. Sometimes there are constraints. There's this whole idea in humans that bipedality, which constricts the shape of the pelvis, is going to limit the size of the skull, and that has all kinds of facts about when human beings are born and the stage of their brain growth at that point. But yeah, I mean, there's a whole there's several literatures on the density of neurons, the ratio of neurons to glia, as well as the relative sizes of different more structures, cerebellum versus cortex versus cortical regions. Your second question is about,
does your work make some statements about
yeah, so I mean the comparative work I've done has been in about 10 or 12 mammals, and we were looking at more specific questions. So we're looking at, for example, New World monkeys versus Old World monkeys, and the postparietal cortex, and how that relates to manual dexterity. And then you choose an animal that's sort of interesting outlier. So you choose the Cebus monkey, which has as good or better manual dexterity compared to Old World monkeys like macaques, and we are a derived old world monkeys. And they are, Seamus monkeys, New World monkeys from South America are so much of an outlier compared to other New World monkeys that there was this theory that they had rafted to South America from Africa, and really that they were a sort of crypto old world. That turns out not to be true. They just independently derive this behavior, and by the way, particular brain features that are very similar to old world monkeys. So it's like they converged on this same neural solution to manual dexterity. So that's the level of kind of question that we're asking. But a lot of other people have written again more generally about looking at overall brain shape in embryos and how they vary across a very large number of species. Where I'm just mapping one or two species.
I can just follow up, just a quick comment. I'm hoping somebody I sent an email to my lab. One nice thing about having my lab controlling this thing is I can tell them to do stuff for Zoom, and I don't have to do it myself. But I'm not doing it yet. But anyway, I'm going to mention a book that's pretty good about some of these topics called Brains Through Time. And it's sort of a combination of a more sort of lay audience book, but also written that can really give you some information about this confluence of evolutionary pressures, environmental pressures, and genetic determinants of how the system has evolved. It's focused exclusively on vertebrates, so it's actually a good complement to what you're talking, and hopefully it'll show up in the chat window in a moment. Quite good in that content. Brains Through Time. I didn't write it, so I get no right.
Keeping an eye on the time, but we do have time for one more question. So I'm going to take a question over here.
Yeah, I was just wondering, you noted that motor cortex seems to be especially variable across individuals, and it has this ability of, yes. Anyways, on that, do we know why that is? Is it something to do with motor neurons, or like the system, the network as a whole? Is it in the genes of these neurons?
I think probably it's to do with what is driving cortical. So in somatosensory cortex, it's pretty nice somatotopy. The homunculus, the sensory homunculus, even though it's distorted, looks is shaped a lot more like the relevant human in the textbooks, or the ratunculus or the batunculus. It's kind of shaped like that animal's skin. The problem with a motor homunculus or a motor mount is that adjacent muscles are not necessarily related to each other. If you're coordinating the hands like this or the hand in the mouth, those are not adjacent places in the body. Depending on what the behavior is, you're going to get different combinations of the muscles that need to work efficiently together. And so there's a coarse somatotopy that's laid out in a similar way to sensory homunculus, where you go from hind limbs to trunk to forelimbs to base. But it's as if you put a blender into that. So fine details are mixed up. So it's often referred to as mosaic or fractured. And so there may be other factors like these behaviors that you identified, where there's a combination of forelimb and face that is a feeding or a defensive defense, or a combination of limbs, all four limbs, and you stimulate them like this. So the resulting map may be some compromise between a coarse somatotopy plus these sort of behavioral clusters that are going to really depend on the behavioral ecology of the animal. Although things like defense are going to be common across all species, and some other things. And then there's even been a modeling study that my graduate advisor did, where he sort of threw these things together and got a weird fraction map that looked possibly like a motor map. So anyway, my speculation, I think that of others, is that one of the reasons you get this variation is because there's a lot of ways to solve this. Whereas there's sort of one optimal or just a few optimal solutions for the sensory map. Maybe that's why motor cortex is so variable and why it's a good model for variation.
Okay, so Victor has convinced me to let one extra question slip in. Victor, you have the floor.
So many people to please try it, it works. Just your last comments reminded me, it's actually this mapping is not just a spatial, but it's actually a spatial temporal situation that we're talking about, because if I remember correctly, when you did the experiments with Michael Graziano, the stimulation time that you applied actually in order to evoke them, was quite long, and that was one of the half a second or something like that. Half a second, which is an enormously long time for actually stimulation. At that time, one of the key criticisms was that it's not just an activation of that patch, but that you're actually activating networks, projections into other areas, and that it's actually network phenomenon and the spatial temporal dynamics that is coding for the actually executed movement. So we're talking about manifolds, of course, dynamics or manifolds, etc. Has there, from my perspective, it's actually not a criticism. I find this fantastic, and I have cited your work very often as an example why more complex, more dynamic objects need to be considered for coding of this type of movement patterns. Has there been any evolution or development of this way of thinking in terms of mapping on spatial temporal network dynamics of these motor primitives, if I may call them motor primitives? I've not followed this work. Is there any continuation in this regard, the deeper understanding in the spatial temporal context?
Yes, so the criticism that we're stimulating and we're activating the network. Our reviewer said that. It was really hard to get this published at first. The reviewer said that, and our response was, of course it's going through a network. What else can it do? It's not going to just magically call up the muscle. It's going through the whole motor system. The point is, our point was, you stimulate, you get something that looks like real life. You show that it's actually necessary for real life. That's the defensive GABAergic studies. And when you ask, like, has this evolved? I guess my thinking is the way I interpret it.
Space and time.
Space and time.
Coding, spatial temporal coding. This is the direction I wanted to.
Yeah, I mean, in that sense, no. I mean, not a lot, except that there's a lot of other people that are doing this now. I think it's still controversial. There's some people that for the reasons you cited, don't think that it's found sort of a heavy phenomenon, which I disagree with. But I think that's one of the things that I want to get with this ENG work is to look at the temporal structure of these. These aren't even motor primitives; these are just muscle primitives, where we're looking at individual muscles that are activated in large combinations from this site, even with very small currents. In contrast to what I was doing with Mike Graziano. One of the many things that I want to look at is that temporal structure. You lose when you make this, what I call a kinematic map. This just says like, well, this body part moves in body part. You lose what we sometimes see, which is the arm will go here and then here, or it will go up and then turn, or you'll get this arm and this arm. A lot of these do look like primitives, but if we can start to really look at the certain elements of those, I'm hoping that will tell us more about how these the fully ecological looking units, how those are actually built. Thanks.
Great, thank you so much, Dylan. That was such an interesting perspective.